In this activity, you will examine leaves that have been modified to perform functions other than or in addition to photosynthesis. While most leaves are adapted primarily for light capture and gas exchange, some plants modify their leaves for support, protection, and storage. These structural changes allow plants to survive in challenging environments or grow in competitive habitats. All of these structures originate from leaves, but their form has been altered to serve different purposes. In this activity, you will focus on three major types of specialized leaves: tendrils, which help plants climb; spines, which protect plants and reduce herbivory; and storage leaves, which store water and nutrients, especially in dry environments. Once again, this illustrates the principle that plant organs can be modified to meet environmental and ecological demands. Tendrils are thin, flexible structures that coil around supports such as fences, stems, or branches. In many climbing plants, including peas and beans, tendrils are modified leaves or parts of leaves. Their ability to sense touch and coil tightly allows the plant to climb upward toward light without investing heavily in thick supportive stems. Spines are hardened, pointed structures that develop from leaves. They are composed mainly of thick-walled sclerenchyma cells, making them tough and sharp. Spines protect plants from herbivores and also reduce water loss by decreasing surface area and creating shade around the stem. In cacti, spines arise from the same position as normal leaves and have an axillary bud at their base, confirming their leaf origin. There are other pokey parts that come from different parts of the plant. A prickle is a sharp outgrowth from the epidermis or bark, and a thorn is a stiff, woody, modified stem with a sharp point. In succulents, leaves are modified for storage. These storage leaves are thick and fleshy due to large parenchyma cells that hold water and dissolve nutrients. They often retain their photosynthetic function, but their primary role is survival during drought. A thick cuticle further reduces water loss. Bulbs, such as tulips, garlic, and onions, are fleshy bases of leaves that stores food underground. In addition to tendrils, spines, and storage leaves, plants have evolved many other specialized leaves that perform unique functions. Bracts are modified leaves associated with flowers or inflorescences. They are often brightly colored and help attract pollinators, as in poinsettias and bougainvillea, where the showy petals are actually bracts rather than true petals. Adhesive discs occur in some climbing plants such as Virginia creeper. These are flattened, sticky structures at the tips of tendrils that allow the plant to adhere tightly to walls, rock, or tree bark, providing strong support. Reproductive leaves are leaves capable of producing new plantlets, such as in kalanchoe. Small buds form along the leaf margins and can detach, root, and grow into new individuals. In this case, the leaf functions directly in asexual reproduction. Phyllodes are flattened petioles that take over the role of the leaf blade. This modification is common in some Australian acacias. The true leaf blade is reduced and the petiole becomes broad and photosynthetic, helping reduce water loss while still allowing photosynthesis. Together, these examples highlight how the basic leaf structure can be transformed to support reproduction, attachment, defense, nutrient acquisition, and water conservation, once again demonstrating the close relationship between structure, function, and environment in plant anatomy. Before observing the specimens, think about how different environments create different challenges. Why do some plants modify their leaves into structures such as spines, tendrils, or storage organs? Consider factors such as herbivore damage, water availability, competition for light, and mechanical support. Record your prediction in your lab notebook before continuing. Examine the plant specimens provided on the next few slides. In your lab notebook, sketch each one and label the type of modification, the plant species, and important structural features such as thickness, rigidity, surface texture, and where the structure arises on the plant. Focus on how the form of each leaf reflects its function. Here we have a common one in Florida, soap aloe. Focus on the fleshy part of the leaf. Despite appearances, the margins of the leaf are not true spines. They are marginal teeth. Pause the video to sketch the leaf. This one is prickly pear cactus. Despite appearance, the fleshy part of this plant is called a chlorophyll, which is actually a fleshy stem, not a type of leaf. In your lab notebook, sketch one leaf of this plant and label the type of modification, the plant species, and important structural features such as thickness, rigidity, surface texture, and where the structure arises on the plant. Focus on how the form of each leaf reflects its function. Pause the video to sketch the leaf. This one is balsam apple or bitter melon. You may have seen its striking fruit. This is an invasive vine in Florida, often smothering native plants. Its stems can grow up to 16 feet long. The foliage is quite stinky when crushed, although it's been used for medicinal purposes. The fruit are toxic. In your lab notebook, sketch one leaf of this plant with its associated modification on the opposite side of the node and label the type of modification, the plant species, and important structural features such as thickness, rigidity, surface texture, and where the structure arises on the plant. Focus on how the form of each leaf reflects its function. Pause the video to sketch the leaves. Specialized leaves demonstrate the remarkable flexibility of plant organs. Whether coiling around a support, forming sharp defensive structures, or swelling with stored water, leaves are shaped by environmental pressures and functional needs. These modifications reinforce a central theme of plant anatomy. Form follows function, and function reflects environment.